based off our previous results? It's a great question.
Okay, thank you.
Okay. I guess that's, yeah, we're right on time. So thank you again, Dr. Schmidt. So let's move on to our fourth finalist today, Dr. Xiufang Xin from the Center of Excellence in Molecular Plant Science of Chinese Academy of Science in China. And she's going to talk about her study on the plant microbe interaction in the phyllosphere. Okay, so Dr. Xin, please.
Can you hear me clearly?
Yes, we can.
Oh,
Hello. I think you haven't played it yet.
okay. Yeah, there was a message saying there's some error. So just want to make sure it's still connected.
Yeah. You're still connected. Oh, not now. Yeah, you're back.
All right. Yeah. Somehow there was a message.
Yep, it's playing.
Okay, good.
Yep.
All right. Hello, everyone. Good morning, afternoon, and good night. So my name is Xiufang Xin. I'm from the CAS Institute in Shanghai. So again, I want to thank the selection committee. It's a great honor for me to talk about our work today. So I think we don't. Plants actually can face multiple challenges during their lifetime. So we have just heard Dr. Francesco talk about how plants sense the oxygen level, right? So obviously there are many other factors, both biotic and abiotic factors, or non-optimal growth conditions that plants need to face. So accordingly, a great portion of the plant genome is actually devoted to sense the environmental clues and to activate downstream responses to help plants adapt these adverse conditions. So I work with plant microbe interactions. The plant diseases caused by a great variety of microbial pathogens in nature is a great threat to plant survival. And there have been statistics showing that a substantial percentage of the yield loss of major crops in the world can be attributed to pest infestation. So it's a big problem. Since my PhD, so I was trained on the plant pathogen interactions, mainly using the Arabidopsis thaliana, so monosyringe patho system. So this is a model system that has been widely used in our field over the last 20 years also to understand the plant resistance and the microbial pathogenesis principles. So this is a gram-negative bacteria that can is mainly used as a foliar pathogen in the lab. So it can enter the plant tissue through natural openings such as stomata and multiply inside the plant leaf in the apoplastic space between the mesophyll cells. So I was trained, my PhD and the postdoc was focusing on the microbial pathogenesis mechanism, or in other words, how do the pathogens cause disease? So for gram-negative bacteria, including the monosyringe I work with, one key virulence weapon is called type 3 effectors, which is a class of virulence proteins that are translocated into the plant cell from the bacteria through a specialized secretion system. So for the bacteria I work with, it generates 36 effectors. So my postdoc work, through my postdoc work, I discovered that this P. stringy bacteria can use two conserved effectors called AvrE and HopM1 to create a water-rich living environment in the leaf. And this is what we call water-soakin. So you can see here, this dark spots basically represent the water-soaking spots in the leaf infected by Xiufang Xin bacteria. So this also occurred in the natural host tomato. And if we mutate AvrE and HopM1, we can see this visible water-soaking will be abolished. So so we know when plants grow normally or in the normal apoplast, there is actually a minimal amount of water, so it's largely filled with air, which is presumably very important for physiological processes such as gas exchange and photosynthesis to occur. So this somehow, we hypothesize that this dry apoplast is actually non-optimal or is an adverse condition for bacterial growth. And pathogenic bacteria such as Xiufang Xin DC3000 can use various factors to drive this water-soaking formation. So this water-soaking could lead to a lot of downstream consequences. For instance, it could make the nutrients more accessible for bacteria to uptake. This actually is proven by a recent paper by David Mackey's group on Nature Cell Host and Microbe. And also it could possibly lead to dilution of the defense compounds by from plants. So which all of this could lead to promotion of bacteria multiplication in the apoplast. And importantly, this water soaking requires high air humidity to maintain, because under low humidity, the water would evaporate from the stomata. So from this work, I just summarized, we found out that we proposed this new model of phyllosphere microbiology. So many previous studies before this study showed that the effectors could suppress plant immunity, which makes a lot of sense. However, through this study, we found that suppression of immunity is not sufficient for pathogenesis to occur. We found that this pathogen-driven wet apoplast is actually another important element of bacterial pathogenesis. So if we make these two processes happen, then the basic pathogenesis can occur. And also, so there's a very famous principle called disease triangle principle in the field of plant pathology, which describes that not only the host immunity and the pathogen virulence are important, but the environmental factors are also very important for the outcome of a plant disease. So it's long observed that the high humidity could promote many diseases to occur in nature, but the molecular basis was not known. So here we, through the funding of this water soaking and the high humidity requirement for this water soaking maintenance, this study provide an important insight into the long-standing humidity influence on disease. In addition, through this study, we also made an observation that in the plant polymutant, in which plant basal immunity and the apoplast water pathway are mutated. So in this polymutant, we found out that not only the basic pathogenesis can occur, but also the bacteria microbiota that living inside the plant leaf can also over-proliferate. So I'm going to mention this later, but I just want to show you that through this, through this work, we also extend or lead me to the area of plant leaf microbiota control. So, so I came back to China to start my own lab in 2017. So I borrowed this from, I think it's my job interview PowerPoint. So the plan at that time was to extend my training in disease susceptibility and work on the plant pathogen humidity interactions, as well as plant leaf microbiota interaction. So the specific questions are listed here. So some of these projects went well than others. But interestingly and also excitingly, we also made an unexpected funding on plant immunity. So now we are also working on plant immunity field. So now we are working on both Arabidopsis and rice as representative of dicot and monocot. So I'm going to tell you what we have learned over the last almost 5 years in these 3 directions. So first, I have told you that we found out the bacterial pathogen can take advantage of the high humidity to generate water-soakin and promote infection. We propose that plants has so what we call the water-soakin pathway, which normally would keep the apoplast water to a minimal level, and therefore plants can grow normally. However, during pathogen infection, virulence proteins such as AvrE or HopM1 can target different components in this pathway and lead to water-soakin. So characterization of this pathway can be important for understanding plant pathogen interactions, because water-soakin is actually widespread in plant disease caused by many different types of pathogens. And in addition, we also think that this study could help address a fundamental question in plant biology, which is how is the apoplast water amount is controlled or regulated. Then we took 2 approaches, which we think this is a fundamental question, but people never ignored or people never studied this before. So we took 2 approaches. First is to use the monitoring effector AvrE as a probe to study which plant proteins are involved in this pathway, and the other is a genetic screen. So I'm telling you, so we recently finished this study and found out that AvrE can actually interact with type 1 protein phosphatase from Arabidopsis. So type 1 or PP1 family is relatively less known compared to, for instance, PP2 families in plant biology. So we found out that AvrE can interact with all the top members when expressed in plant top or in vitro protein assays. So Arabidopsis top has 9 members. So we actually used CRISPR technology to knock out multiple tops and generate different lines of polymutants. And finally, we found out that one line, the top 12537, showing very interesting the spontaneous water-soakin on the high humidity. So this is without pathogen inoculation. And furthermore, when we infiltrate with the bacterial pathogen, the pathogen-driven water-soakin was also more severe in this mutant compared to the wild type. And this is accompanied by higher bacterial growth in this mutant. And then in addition, we wonder why this mutant show this interesting water-soakin phenotype. And then we found that this is a mutant actually show ABA hypersensitivity. So here I'm just showing you one of the assays for ABA sensitivity. We found that the top mutant, for instance, has a very strong stomata closure phenotype. So this result suggests to us that the ABA pathway and possibly stomata movement are involved in the water-soakin formation. And indeed, so I'm showing you that when we do bacterial inoculation using wild type strain or AvrE HopM1 mutant strain, we see that this bacteria can induce ABA responses, including ABA accumulation, gene expression, and stomata closure in an AVRE HopM1 dependent manner. So for instance, here, this result induced to a very high level of ABA, but AVRE HopM1 induced to a much lower level. And there's the same trend for the expression, and also this result induced a very strong stomata closure, which is abolished in the AVRE HopM1 mutant. So without further details, so from this study, we found that the P-swingy effector AVRE targets the Arabidopsis top SRK module to activate the ABA signaling and induce water-soakin. And also, this study also suggests to us that the plant ABA pathway and the stomata aperture are actually important for regulating the apoplast water status in the leaf. So our study was also supported by another study from Dr. Peter Murphy's lab that this paper came out in the same issue. So they mainly worked on the effect HopM1 and found out HopM1 may adopt a different strategy, but also to achieve stomata closure and water-soakin. And also, interestingly, there's a very recent paper that came out in Nature showing that actually plants also evolved this small peptide called screw, so which can be recognized in plants and leading to reopening of stomata and promotion of water loss in the leaf. So again, I think this together with our studies highlights the importance of stomata aperture control and the apoplast water regulation during pathogen infection. So this is our study on pathogen and humidity interaction. What about the other direction, right? So does high humidity also affect plants, particularly host immunity? So I already told you that this important disease triangle principle, and also the high air humidity promotes many plant diseases in nature. However, the molecular basis for whether and how humidity affects host plants was poorly understood. So we initially performed an RSPS experiment using Arabidopsis inoculated with Pseudomonas aeruginosa bacteria and found out and placed the plants under different humidity and found out that interestingly, there are many immunity-related genes such as jasmonic acid or salicylic acid, which are important defense hormones. So genes related to these pathways are differently regulated by humidity. However, one problem is that the pathogen infection will actually activate multiple immune pathways, including the SAJ, S-link pathways, and these pathways can crosstalk with each other. So it's very complicated. We therefore took a strategy of treating plants with different elicitors to activate individual pathways and try to answer when does high humidity affect specific plant immune pathway, and then second, does high humidity affect dicot and monocot plants in the same manner or via the same mechanisms. So it actually took us quite some time to set up the system. And I'm just showing you that we found out in Arabidopsis, the salicylic acid, which is a very important hormone, especially for defending against biotrophic or hemibiotrophic pathogen in Arabidopsis. So SA pathway is dramatically inhibited by high humidity. So here, BTH is a analog and can induce SA responsive gene expression. We can see this expression is dramatically suppressed, and also the SA level is significantly suppressed by high humidity. Interestingly, in cultivar rice, we found out the salicylic acid level is not obviously affected. However, another very important defense pathway in rice, the jasmonic acid pathway, is significantly inhibited under high humidity, as shown here, and also the JA level. So this tells us that high air humidity condition actually puts plants under an immune compromised status, which can make plants vulnerable to pathogen infection. So we are currently working on the molecular mechanism and try to identify the components that are directly affected by high humidity in these pathways and in these different plant species. I'm just sharing you with one piece of data that in Arabidopsis, we found out that one central SA signal transduction element, the NPR1 protein, seemed to be affected by high humidity. We found out that the nuclear localized NPR1 protein accumulates more under the high humidity, and this is also accompanied with lower polyubiquitination level of the protein. So hopefully I can update you with a more complete story in the near future. And the long-term goal for this direction is to first uncover the molecular mechanisms of humidity influence on particularly on host immunity. And also in the long term, we want to generate the high humidity resistant plants by genetic engineering of the humidity targets and to hopefully provide novel approaches for improving plant resistance. And because high humidity actually promotes many diseases in nature, we hope the new approaches may offer a broad spectrum resistance. So that's the first part. So the second direction is plant leaf microbiota. So I'm sure many of us have known that the root microbiota has been a very fast approaching area over the last few years. So in contrast, the leaf microbiota or the phyllosphere microbiota was less understood. I already mentioned to you that I started working on this also from my postdoc work, in which we showed that we found out that if we block the basal immunity and apoplast water pathway, this will lead to the overproliferation of the bacteria inside that grow inside the plant leaf or the endophytic population. And interestingly, this is associated with obvious tissue damage, such as fluorosis or necrosis in the leaf, in the mutant. So this tells us that this actually displays a form of dysbiosis in the plant kingdom. So this dysbiosis was first described in human microbiota, which means the microbial imbalances and your link to diseases. So we think this gives us very important implications that plants actually evolved ways to normally maintain a balanced microbiota. So we decided to have a further functional study of microbiota homeostasis and leaf health. So I started initiate this project, but then I came back to China to have my own lab. So the following work has been finished by outstanding leading scholars and postdocs in Shenyang's lab, Dr. Shenyang He's lab, Dr. Tao Wang's lab, and also other collaborators. So we did further a detailed analysis of the bacterial community in the mutant. And interestingly, we found out that this community shift in a way that resembles what occurred in the human bowel disease. So for instance, in both cases, we see the decreased bacterial diversity and also increase in the proteobacteria and reduction in the firmicutes. So this suggests some interesting parallel between the different kingdoms. One important part of this paper is to establish whether there is really a causal role of microbiota homeostasis to plant leaf health. So for this, we generated the synthetic communities from the different plant genotype, and also using the notobiotic system, we showed that indeed a misregulated microbiota, for instance, from the thinkum from the nymphatic plants, can be harmful for the plant growth. So the take-home message for this work is that we uncovered a plant genetic network that can guard or control the homeostasis of leaf microbiota. So if the components are mutated in this network, then we'll see certain, especially the opportunistic microbe will overproliferate and then lead to the dysbiosis phenotype and also harmful cause damage to the plant health. So the last part I want to talk about is plant immunity. So we know we in our field we often say that the disease for plant disease is an exception. That's because plants evolved the immune system to activate defense responses to fight against the pathogens. However, differently from human pathogens, which has adaptive immunity and specialized immune cells, plants only have innate immunity, which means, and we call it a two-layered immune system because it relies on two classes of immune receptors to recognize pathogen-derived, what we call non-self molecules, and initiate signaling pathways. So the first class of receptors is called the PRR, located on the cell surface and leads to PTI. And I already told you that virulent pathogens can use effectors to shut down PTI and cause disease. And some of the effectors can be recognized by a second class of receptors called NOR and leading to ETI, which usually generates strong resistance. So plant immunity has a very long history of study, and immune receptors also particularly have been extensively studied. So one important message is that these two classes of receptors have very different protein domains, domain structures, and activation mechanisms. So here I'm listing PR, for instance, are RLK type proteins, whereas the NOR are totally different. And the early signaling events was also different. However, despite this so many studies, one fundamental question that remained in the field was the relationship between the PTI and the ETI pathway. So although there were indications suggesting the PTI and the ETI share overlapping responses, but a clear relationship between the two pathways was not known. We worked on this with surprising results. So here we used a PR mutant, in which the PTI signaling is lost. And interestingly, we found out that, so we did a disease assay against ETI strain. So this disease-resistant carrying AvrE2 this AvrE2 can be recognized by plant NOR called RPS2 and initiate ETI. So here we can see in co-zero, this mutant strain will grow much less due to ETI resistance. However, in this FEC and BBC plants, the bacteria can multiply to a high level, suggesting that the ETI resistance is compromised. So we also show this phenotype also occurred in ETI triggered by different factors. So of course, the important question is the important thing is to understand why. So we did many assays to find out what immune ETI related responses are wrong in these PR mutants. So here to summarize this response, these are NOR expression or the other early signaling events are intact. But finally, we were able to find out there's one immune response, which is the reactive oxygen species or ROS production actually requires both PTI and the ETI pathway. So here I'm showing you in this AvrE2 transgenic plants, we can use different elicitors to activate different pathways. I'm showing you that, well, so this is the ROS measurement. So FLAG22 will trigger a fast but very transient PTI ROS. DEX alone will only trigger a late and a weak phase of ETI ROS. However, FLAG22 plus DEX can not only trigger PTI ROS, but also trigger a very strong secondary ETI ROS. So this suggests to us that this strong ETI phase of ROS actually requires both pathways. We confirmed this in this DEX AvrE2 in the PR mutant background. And then using different approaches, we were able to show that this ETI ROS is mediated by membrane localized innate pH called abiotic. So here we use a fluorescent dye to label the H2O2. So we can see this H2O2 signal very strongly in the apoplast, and while in the abiotic mutant, the signal is completely lost. So one important piece of information.
Excuse me, Dr. Xin. I'm sorry to interrupt. It's very comprehensive work, but we are already behind the schedule. So could you please try to close up as soon as possible? Thank you.
Okay. Yeah. So sorry about that. So just this one last piece of data showing that this we found out these two pathways can actually coordinate to fully activate abiotic. So one is can upregulate the transcription protein, and the other is for the phosphorylation. So the take-home message for this one is that the PTI and ETI pathway can collaborate and are synergistic to each other. So I'm not going to the further detail. And again, we published this paper side by side with Dr. John's lab, and we took complementary approaches and support each other. And there is also an emerging theme on the intimate crosstalk of PTI and ETI. So, and commentaries. So just to summarize, so I have been also still working on this host pathogen and environment triangle interactions. And our long-term goal is to hopefully offer innovative strategies to improve plant resistance and the phyllosphere health. So I really want to thank the students and postdocs in my lab. It has been great 5 years, and I want to thank collaborators and also the funding source, and especially I want to thank our institute and also the CPAMS, which is the joint program of CAS and the GIC, to offer the generous support for my setting up the lab, and also the great platform for scientific communication and training for junior PIs. So thank you very much. I'm sorry about the timing.